Okay, for today's lecture, we're going to talk about Hardy-Weinberg, the Hardy-Weinberg principle. Okay, so basically this is used to test whether or not a population is evolving. You will have some genetic variation within a population, but that does not always ensure that a population will evolve. So with Hardy-Weinberg, if you do out the Hardy-Weinberg equation, you have your known alleles. You know it will not evolve if your Hardy-Weinberg always equals 1, which it should. If for some reason it doesn't come out to be 1, you know that it is evolving due to breaking those 5 assumptions, and we'll go over those in a little bit. So let's get down to some basic terminology. We have population. So basically a population is a group of the same species of individuals in the same area, and they breed and they reproduce fertile offspring, which is key. Okay, within that population, okay, you're going to have a gene pool. And so with this gene pool, you're going to have all the copies of every type of allele within all those members of the population. So let's do an example. Okay, let's pretend we have 500 birds. Sorry about that. All right, and for those birds, our main alleles, we'll do CR and CW. Remember, every species is diploid, so they're going to have 1 and 1. Okay, and let's say these code for pigment. So let's pretend that we have CR for redbirds. Okay, and we will have CW for blackbirds. And CW for whitebirds. Now this W here, you can think of this as big R, little r. We're using W for this due to it being incomplete dominance, and so it shows the phenotypes a lot easier than if we were to do big R, little r. Okay, so I'm going to move on to slide number 2. Okay, and so let's go back up here. We have 500 birds. Okay, let's pretend that 320 of our birds are red. And so let's remember that it's CR. Okay. We'll do 160 are black. And that would be our CW heterozygous. And then 20 to equal up our 500 are white. CW. And you can easily think of this as homozygous dominant, and this is going to be our hetero, and this one is our homozygous recessive. Now this only talks about our actual population. Let's think about our gene pool. Okay, so in essence we actually have 1,000 alleles within our gene pool, because, again, this is a diploid species, so 2 alleles per individual. What we're going to do for our first allele, CR, is actually going to be 800. Now, where do we get that 800 from? Okay, see if I can change color. All right, so we know that we've got 320 of one CR and 320 of the second CR. So this is going to be 320 plus 320. And then we also have 160 of from the blackbirds. That's going to give us our 800. Okay, 800 over 1,000, and it's going to be 0.8. So remember that number. Let's go on to slide number 3. Okay. So now we want to know CW. Let me pick a different color for that. CW. Okay. Now let's remember that we had our 500 birds, 1,000 alleles. For CRCW, we had 160, so that means that we have 160 CW. And then CW was 20, so we're going to do plus 20 plus 20, which gives us 200, and that's over the original 1,000, which equals 0.2. So if we did 0.8 plus 0.2, we get 1, or CR plus CW equals 1. When you guys do your pre-lab, you are going to see, and this is pretty common, where you've got your one allele plus your other allele is always going to equal 1. From that, you can then use the main Hardy-Weinberg principle to see if it's actually in equilibrium. So keep going. So this is our pretty common, well-known equation for Hardy-Weinberg, and you're probably going to wonder what all this means, and we'll go over this. Okay, so from before, that CR plus CW equals 1, let's call CRP and CWQ equals 1. This here is going to be our CR. This here is going to be our heterozygous, always heterozygous. So this is going to be 2 times our CRCW. And this is always going to be our homozygous recessive. So this is going to be CW. That's going to equal 1. So let's go back to our numbers that we know. So we know that P equals 0.8 and Q equals 0.2. So we have 0.8 squared plus 2 times 0.8 times 0.2 plus 0.2 squared. That's all going to equal 1. And so if all of that comes out to equal 1, we know that it is not evolving. In our lab, we're actually going to be looking at natural selection. So with natural selection, you guys probably went over this in lecture. It's based on different successes and the reproduction of offspring with the desirable trait that was successful. So these certain traits lead to a greater relative fitness, and this relative fitness can actually alter the frequency distribution in 3 different ways. And so you guys are going to explore that while you do the actual lab. And I'm briefly just going to touch upon the 3 different distributions. Okay. Let's pretend, and this is right from your book. Okay, so let's pretend we have our birds again. Okay, so we'll go with, let's say, 5 shades of birds. Okay, and from very, very light, so we'll do a white bird, white bird, all the way up to blackbird. Okay, and then all the 5 different shades in between. So 1 white, 1 black, and then 3 shades of gray. Okay. So if we pretend that our normal distribution is going to be like this, where we have 1 extreme allele, the homozygous dominant, here, and homozygous recessive will be here. Let's pretend that we have a shade here, here, and here. So that's how they're distributed. Okay, with this being, again, original. Okay. Now the 3 different types, and let me just erase this out. It's not going to let me. Okay. Alright. So 3 different types. We've got directional, distributive, and stabilizing. And I'm going to go on to the next slide. So we'll go with 1 directional. Okay, and that is where there are certain conditions, environmental factors that will favor individuals that exhibit an extreme phenotype range. And the key word for this is range. So if this was our original, again, remember, this is going to be our bird in the middle, our 2 birds here. Okay? A directional selection is going to shift like that, where now you've got the curve over these for some reason or another. Maybe the darker birds blended in better with the surroundings and were unable to be picked off by neighboring hawks. And so this caused them to reproduce more, to have more chicks, and you have this directional range. Whereas the lighter colored birds stood out more, picked off easier. Okay, but again, range. Okay, number 2 is distributive. Disruptive. Disruptive. Okay. And this is where there are certain conditions that favor 2 different extremes of your range. Okay, so again, remember your original. And your original instead now is going to go and look something like that, where your heterozygous are not that popular, whereas your homozygous are going to be much more popular. Our 2 extremes, so remember this was a blackbird, no, these were a white bird, and this was a blackbird. So let's say there are white trees and white leafed flowers on the trees that the birds would camouflage in, and so the hawks couldn't get those. And then the blackbirds blended in with some other foliage, but for some reason these gray shaded birds were picked off. Okay, so this would give you a disruptive selection. The last one is stabilizing. Okay. And for that, we're going to need more room. So we're going to go to the next slide. All right, so 3. Stabilizing. Okay, so let's pretend that, okay, so this is our original. It's now going to go to this. Okay, so instead of the range of phenotypes being in the middle, it actually picks out the heterozygous as being the best. So let's say that the gray birds found gray foliage, and the blackbirds and the white birds stood out, but the gray birds didn't. And so the blackbirds and the white birds got picked off by predation, whereas these gray birds thrived, reproduced, and did very, very well. Okay, so those are your frequency distributions. Okay, when we are looking at Hardy-Weinberg, there are these assumptions. I'm going to briefly go over them. Assumptions are, I'll end on this, so because you need to know these assumptions. 1, no mutation. Okay, so in order for it to, for a population not to evolve and to follow Hardy-Weinberg principle, you cannot have mutation. You must have random mating. Okay, no natural selection. So in lab, we're going to break that rule. A large enough population size. So you want a pretty large population size so that you do have random mating. And the last one, no gene flow. So think of your genetic pool, again, all your alleles in your population. You don't want gene flow, meaning genes leaving your population. If you have gene flow, this is going to influence evolution, and it's not going to stay within that Hardy-Weinberg principle.